My name is Marjon Wouters from KU Leuven, Belgium, and I'm going to describe my GXMed paper entitled Dominant Negative ADA2 Mutations Cause ADA2 Deficiency in Heterozygous Carriers. ADA2 deficiency is an autosomal recessive disease with an estimated disease prevalence of 1 in 220,000 individuals, corresponding to a carrier frequency of 1 in 236 individuals. The disease is characterized by a broad clinical phenotype divided into three categories. Heterozygous carriers are defined as individuals carrying monoallelic pathogenic variants and are mostly regarded healthy. The diagnosis consists of clinical assessment, the presence of biallelic pathogenic mutations in the ADA2 gene, and deficient adenosine deaminase activity in serum or plasma. Although ADA2 carriers are mostly regarded as healthy and asymptomatic, several publications reporting typical ADA2 clinical features in ADA2 carriers have been published. In our own experience, we have a cohort of 10 patients presenting with a ADA2 phenotype, but only carry a single variant in the ADA2 gene. However, no mechanistic explanation has been described so far. Therefore, we wanted to investigate which mechanisms are at play. We did this by assessing the effect of ADA2 variants on ADA2 wild type by co-transfecting wild type and mutant ADA2 in a 50-50 ratio to mimic carrier state. Next, we made use of population genetics to assess the correlation between carrier status and phenotypes relevant to ADA2. First, we assess the effect of ADA2 variants on wild-type ADA2 secretion. Since dimer formation is required for of ADA2 is required for ADA2 enzymatic activity, we made use of non-denaturating gels. Here, we observed that compared to wild-type 50%, a small reduction in secretion was observed for variant G47V, and this reduction in secretion was even more pronounced for variants R169Q, H424N, and Y453C. Next, we investigated the effect of ADA2 variants on the adenosine deaminase activity of wild-type ADA2, both intracellularly as well as extracellularly. In heterozygous conditions, certain variants exhibited an enzymatic activity level below that of wild-type 50%, which is suggestive of a dominant negative effect, and this varied depending on the studied variants. Therefore, we made the distinction between an absent, a weak, and a strong dominant negative effect. Here we observed that all variants except for G47W exerted a dominant negative effect on wild-type ADA2, both intracellularly as well as extracellularly. Lastly, we made use of population genetics to assess the correlation between carrier status and phenotypes relevant to ADA2. 0.16% of the general population is a heterozygous carrier of a dominant negative ADA2 variant. Moreover, R169Q is the most common variant in European, Finnish, and non-Finnish ancestry groups with a frequency of 1 in 1,000. And because of this, we assessed the enrichment of DADA2 phenotypes in R169Q heterozygotes, both in the UK Biobank and FinGen. And here we have found an enrichment in several DADA2 related manifestations. To conclude, our in vitro study of ADA2 variants identified in the patient cohorts affect the secretion of wild-type ADA2 protein and exert a dominant negative effect on the enzymatic activity of wild-type ADA2. Moreover, population genetics show that R169Q heterozygotes are associated with DADA2 phenotypes. And these findings imply that the incidence of DADA2 might be higher than the published 1 in 220,000 individuals, especially since the variants described here represent a significant fraction of pathogenic ADA2 variants. And this makes these findings especially relevant for an important proportion of DADA2 patient kindreds. With that, I would like to thank our collaborators and feel free to contact us.